Alright guys, so we'll continue on where I left off. Sorry about the breakup, having more technical issues. So we'll continue on with signs and symptoms and how they present in different parts of the body and so forth. So I think I've already talked about this slide. Signs and symptoms of inflammation. So generally fever, pain, soreness, swelling, all of these things are systemic effects that are associated with inflammation. Edema as a hallmark of inflammation. This is a buildup of fluids. Generally, that's what we consider inflammation to begin with. Granulomas and abscesses, so these are again just conglomerations of immune cells that kind of wall off infectious things or non-self particles, and they can form these nice little nodules. Lymphadenitis is swollen lymph nodes just from the draining of lymphatic tissues and cells into the lymph nodes. And then lesions. This tends to happen at the site of infection or disease. You get stuck in the arm with something like E. coli or some kind of nasty pathogen. You'll get a nice little kind of swelling that occurs in that area, and you'll tend to break down tissue in that area because that's where they're the most concentrated, at least initially, and that's when you'll get a lesion. Signs of infections in the blood. So you'll get change in number of circulating white blood cells. It's either leukocytosis, which is an increase in the number of white blood cells, or leukopenia, which is a decrease in the number of white blood cells. Depending on what the cells infect and what they do to the body, that determines what type of change in the white blood cells will occur. Leukopenia is most commonly found in things like HIV. That's because they target white blood cells. But leukocytosis you'll find in a lot of bacterial infections because you're increasing the number of white blood cells to ward off infection. So septicemia, this is a general state where microorganisms are multiplying in the blood are present in large numbers. So septicemia is where you actually have microorganisms in the blood and they're multiplying. So this is very severe. If you have microorganisms multiplying in the blood, you are in serious trouble. And that is because they can pretty much get as much nutrients as they want from the blood, and it allows them to travel throughout the body fairly easily. Bacteremia or viremia, so these are microbes that are present in the blood but are not necessarily multiplying, so it's kind of a transitional state. Septicemia is much more severe than bacteremia. So portal of exit, like I've mentioned before, this is very important in continuing the life cycle of a pathogen. Here are some of the more common portals of exit. Coughing and sneezing, that's an obvious one. Cells or skin cells or open lesions, not as common, but it is present. Urine, especially in sexually transmitted diseases. Feces in the fecal-oral route. Removal of blood in a lot of bloodborne pathogens. And insect bites. Insect bites are also very important, especially for tropical diseases like malaria. So exit portals. So most commonly it's the respiratory and salivary glands, and that's because we're constantly releasing saliva, we're constantly opening our mouth, we're constantly coughing, we're constantly sneezing. So it's an easy way to get out. Skin scales, not nearly as common, but there are some organisms that prefer to live right below the skin surface, so it's easy for them to kind of slough off with the skin scales. Fecal exit, this is very common in the fecal-oral route, so a lot of your gastrointestinal disorders are transmitted this way. Your genital tract, all of your STIs are transmitted this way. And then removal of blood or bleeding, so bloodborne pathogens fit in this category. So microbes, if you're a good pathogen, you need, like I mentioned before, you need to be able to persist in your host of choice without necessarily killing your host. And one way of doing this is by becoming latent. And latent is where you kind of have a period of time where you're not really infecting or causing damage to the host. Instead, you're just kind of hanging out, low numbers, waiting for a good opportunity to arise for you to kind of wake up and take over the body again. A lot of viruses are very good at this, so herpes simplex 1 and 2, hepatitis B, AIDS or HIV is very good at this, and Epstein-Barr. So A lot of these viruses do this by incorporating their genetic material into the host, but then kind of shutting off or having mechanisms that shut off the transcription and translation of that viral DNA. And by doing this, they kind of just hang out in the cell in a resting state. This allows for any time that that cell multiplies normally or divides normally, it kind of expands the amount of the virus present in the body. And then some kind of response or some kind of stimuli occurs and it kind of wakes up the virus, and then it becomes infectious and starts doing its job. And then a sequelae is a long-term or permanent damage to tissues or organs. So generally, sequelae is kind of thought as a consequence of having a disease. So reservoirs, so this is the primary habitat in the natural world where pathogens originate. So being a good pathogen, you need to be able to survive in the environment as well, because that's where you're going to be taken into your host. You don't have to survive, you don't have to multiply extremely fast or happily, but you have to be able to survive. So there are living reservoirs. They're generally considered carriers. You have asymptomatic carriers. These are people who do not exhibit symptoms, but they still carry the organism. You have incubation carriers. These are where the organism kind of incubates, grows, and divides, but doesn't necessarily make the person sick, but they can then transmit it on. You have convalescent carriers. These are people who are getting over the disease. And as they're getting over it, they're still infectious. You have chronic carriers. These are people who are always carrying the organism, but don't always explain or display symptoms. And then you have passive carriers. These are people who transiently carry the microbe. It's not actually growing and dividing in them, but they get it, they transmit it, and then it goes away. So here's a nice little example of all of these in a picture format. So animals as reservoirs. We have vectors. So this is a live animal that transmits an infectious agent from one host to another. A good example of this would be mosquitoes. A lot of these are insects that like to land on people. And there's two different types of vectors. You have a biological vector, so this is where part of the pathogen's life cycle is associated with the vector. So generally it's internal. Mosquitoes are a good example of this, tsetse flies, ticks. All of these guys have microbes that utilize the structure and the function of the tick or the ectoparasite or the mosquito and take advantage of that to transmit the organism. Mechanical vectors, on the other hand, just carry microorganisms that could be pathogens on their body, and then when they land on something or they brush up against something, it transmits the organism that way. It doesn't actually go internal, and it's not necessarily a part of the life cycle of the pathogen. So here you can see, here you can see a mechanical vector. So generally flies like to land on poop. It's where they lay eggs. They can carry around E. coli in their legs and then transmit it to subsequent people or subsequent animals or whatever the case may be. Mosquitoes, on the other hand, actually actively take up blood, and in that blood you can find things like malarial parasites, and those malarial parasites actually utilize the digestive tract of the mosquito to continue out its life cycle and transmit the malaria. And this thing doesn't want to advance to the next slide. Okay. So zoonosis. This is an infection that's typically found in animals, but has been somehow transmitted to humans. Whether that's through mutation in the pathogen or just the fact that we've come into close contact with animals we've never seen before. But this allows for kind of a transition of an animal pathogen into humans. Most really good pathogens that we see today were originally found in animals, and they are zoonoses. Things like HIV, influenza, hepatitis. These are things that were found in animals, SARS, and then they were transmitted to humans. And generally, it's happening quicker and quicker as we evolve. So if you look in like the 1900s compared to now, there's a lot more zoonoses now than there were then. And it has a lot to do with the fact that we're spreading ourselves out as a human population. So we're going into forests, we're going into areas that we haven't been to before, and we're becoming exposed to different types of animals that we've never seen before, in close contact with these animals. And that allows for these kind of pathogens that are associated with them to kind of transition into us. That's how HIV started, that's how SARS started. But it can be somewhat problematic. Excuse me. So here are a few common examples. For rabies, you can see, or viruses, you can see rabies, yellow fever, viral fever, hantavirus, influenza, West Nile virus. All these guys you've heard about before. For bacteria, things like plague, salmonellosis, anthrax, all found in animals first and then transmitted to us. And parasites are notorious for this. Almost every parasite has an animal reservoir of some sort. So non-living reservoirs, this can be the soil, it can be water, it can be an environment, it can just be anywhere the bacteria or pathogen needs to hang out before it can infect someone else. So how are things transmitted, or are these pathogens transmitted? You can have a communicable disease. This is the vast majority of them. It can be transmitted from one host to another host, whether that's through saliva in the mouth, through blood, through urine, through feces, whatever the case may be, it's still transmitted directly or indirectly from one human to another. Non-communicable diseases does not arise through transmission of the infectious host from a host, but it's acquired through some other circumstance, whether that's becoming compromised, immunocompromised, and your normal flora actually become pathogenic, or you acquire it from a non-living reservoir and you don't really know where it came from. So maybe I got influenza from licking a doorknob. Well, I probably got it from someone who had influenza, but I can't make that connection directly because I'm not 100% sure, so it's a non-communicable disease or non-communicable route of transmission. So patterns of transmission, so this is pretty much common sense. You can see contact, droplets, vertical, so from mother to child. Biological vectors such as mosquitoes or ticks or whatever the case may be. Can also be from fomites, bacterial water. Fomites are non-living vectors. And then air is also obviously a way of transmitting organisms. So you can have contact transmission or indirect transmission. So indirect transmission uses some sort of vehicle of either transmitting the organism or allowing the organism to kind of hang out and wait for a good host. There can be things like fomites, again, which are inanimate objects that hold pathogens. Food, water, biological products, be the air, whether it be aerosols from sneezing. So when you sneeze, you shoot out aerosols a good 20 feet, and then they can kind of float around. They're not heavy enough to fall to the ground, but they just stay lofted in the air. That's how things are transmitted. Or droplet nuclei. These are the larger water droplets, which actually fall to the ground fairly rapidly, but they carry a lot of organisms with them, so they do become fairly infectious. So direct contact, it's touching, kissing, sexual intercourse. Perinatally, so that's crossing through the placenta, or perinatally, that's during or shortly after birth. So HIV is transmitted perinatally, but toxoplasmosis is transmitted prenatally. There's this nice there's the droplet nuclei we talked about, and then you have these going far out this direction. You have all of these aerosolized particles that contain infectious agents. You can see this guy's from 1940. It's a very famous photograph. You'll see it in almost every microbiology textbook. Lastly, what are nosocomial infections? So these nosocomial infections are infections that occur in a hospital setting. Typically, these can be these infections tend to target people who are immunocompromised, and whether that's because hospitals are full of immunocompromised people or the pathogens have evolved into affecting immunocompromised people is yet to be determined. But because you're putting a lot of sick people in one area, and if you're a hospital that isn't very good at warding off infected patients, you can run into a lot of problems. So if you have immunocompromised ward in one portion of the hospital, then right next door you have the infectious disease center. You can see how that can be somewhat problematic. You're going to basically make people who are very susceptible to getting pathogenic infections come close to pathogens on a fairly regular basis, and you'll run into problems. So there's a lot of good hospital practices that have come about to kind of make sure that these things don't happen as often. But nosocomial infections are a breeding ground for evolution in microorganisms, especially when it comes to resistance to both chemicals and antibiotics. And that is because they're under constant natural selection pressure. Constant pressure of using either the chemicals to disinfect everything because hospitals are notoriously clean, or you're surrounded by patients who are on multiple cocktails of antibiotics. So it's kind of a nice breeding ground for these resistant microbes, and for that reason you get some nasty nosocomial infections such as MRSA, which is methicillin-resistant Staphylococcus aureus, which is almost resistant to almost all the antibiotics that we currently use, and it likes to infect skin and cause necrosis or dead skin, open oozing sores, which is pretty disgusting. So there are about 2 to 4 million cases per year, and it's important to follow medical asepsis, so sterilizing, keeping things clean, and making sure that if you are using a chemical to disinfect, that you use it to its full extent and you disinfect everything, don't leave anything behind. You can see the vast majority of urinary tract infections, and that's because a lot of people get catheters, and catheters tend to get biofilms around them, and it just is a breeding ground for pathogens. But you can see respiratory, surgical sites, meningitises, gastrointestinal, skin, septicemia. You get a little flavor of everything. So, universal blood and bodily fluid precautions, or universal precautions. So this is a guideline from the CDC on a way to prevent illness spread from blood and other bodily fluids. So the biggest thing from this is to assume that all patient specimens are infectious. Just because you know someone or you know that they're not infectious, don't treat their blood any differently than someone else. Just treat it all the same, and then you have nothing to worry about. So what causes a disease? It's called the etiological agent. That is the organism that has caused the disease you see in a patient. And sometimes it's easy, sometimes it's hard to determine. When it's hard to determine, that's when it becomes difficult to treat the patient because you don't know exactly what to target. So the first development in determining the etiological agent is Koch's postulates, which we've talked about before. This kind of developed a standardized method, but the problem with this method is that it uses phenotypic analysis, and it also requires the organism to be culturable, and not all organisms are culturable. So we've talked about this before, so find evidence of a particular microbe in every case of the disease, isolate that microbe from an infected subject and cultivate it in pure culture in the laboratory, inoculate a susceptible healthy subject with the laboratory isolate and observe the same resultant disease, and then re-isolate the agent from the subject. So it's a long process, and again, it requires the ability to culture the organism. Also, there are some ethical issues here, so if it's a disease that's only capable of infecting humans, like HIV, do you re-inoculate a healthy human with your causative agent to see if it is HIV? Probably not. So there's ways around Koch's postulates, such as doing genotypic analysis, so sequencing, or maybe doing PCR, looking at the actual DNA, the genes that are associated with the organism, make it much, much easier, much, much quicker, and less ethically problematic. So here's a nice little picture if you're interested. So quickly about epidemiology, I'm not a huge fan of epidemiology, but I do need to talk about it a little bit. What epidemiology is the tracking of diseases geographically. So looking at where disease starts in maybe in a country or a city or wherever, and then watching it spread and trying to determine what's allowing it to spread and how to prevent it. So epidemiology is the study of occurrences of diseases. So an epidemic is a sudden acute disease outbreak affecting many people. So it's within a localized area. A pandemic is a worldwide epidemic. And then an endemic is a chronic occurrence in a geographical region. So epidemics are sudden and they're acute. So it could be things like a foodborne illness or something from eating infected vegetables in that area. A pandemic would be if those vegetables were transmitted throughout the world and everyone got sick with that same pathogen throughout the world, no boundaries. And then an endemic is something like a cold that happens every single year. It's not sporadic, you know it's going to happen, and it's within a specific centralized location. And then sporadic are just kind of random outbreaks, and these can be the hardest to determine because you're trying to figure out why one's happening here, why one's happening here, why one's happening here, and sometimes there's no real connection. So here's an endemic occurrence. So it happens fairly regularly in these known areas. We know it's going to happen. Here's epidemics. So they're sporadic, focal in their location, but they can be spread out. Sporadic, small cases, kind of all over, no real pattern. And then a pandemic is just global. And this is because of travel. The ability to travel around the world within 24 hours makes it very easy for things to spread. Okay, come on. Next. Okay. So who, when, and where the tracking disease in the population. So epidemiologists are concerned with virulence, portal of entry, portal of exit, and the course of a disease. And they are interested in surveillance. So there's this whole kind of system set up for reportable diseases. So by law, there are a handful of diseases that must be reported to the CDC every single case that is received. And that allows the CDC to kind of track where these diseases are migrating, where they're coming from, and ways to prevent that. So methods of epidemiology, so you can get sources of information either from public record questionnaires, surveys, hospital records. There's a little bit of this information almost everywhere. It's just how you utilize it, how you put your resources together to fully extract it. You also have notifiable diseases, so these are diseases that are required by the CDC for doctors to let the CDC know that they have a case of this disease. And they're generally things that are somewhat serious, also can transmit or be transmitted fairly easily, like HIV is a notifiable disease. Now the doctor doesn't have to say, okay, so-and-so has HIV. They don't have to give any of the patient information. They just have to say, I live in this community and this person has HIV. I need you to track it. And then they look at the statistics of this. So you look at the incidence rate or the prevalence rate. So the incidence rate is how many cases you have in a specific area at any given time. The prevalence rate is how that is going to change over time. So how much more you have from point A to point B, or how much less you have from point A to point B. Where the incidence rate is how much you have at any given time. It's like a snapshot. Here are the reportable diseases in the United States. So you can see things that are spread fairly easily, like gonorrhea are included. But you also see things that are pretty nasty and that we don't really see too often, but when we do, you need to report it, like rabies, for instance, or plague. So how do you measure infection? So there are some experimental procedures with animal models, but again, this can be problematic because you're looking at an animal model against a human model, how similar, how different they are, and all that good stuff. But sometimes it's the only thing you have. So you're looking at the infectious dose or the morbidity. So how much it takes for the animal to become sick. So morbidity is kind of the yicky feeling, being sick, not being able to work, not being able to do this or that, but not necessarily death. The ID50, so this is the number of cells required to infect 50% of the subjects. And then there's the lethal dose, or the mortality, the LD50. So this is the number of cells required to cause death in 50% of the subjects. So morbidity is just icky feeling, sickness, general blah, where mortality is death. and we're done. So enjoy. Bye.